Okay. Um, Good afternoon, everyone. My name is Andrew Van Dusen, and I'm one of the summer trainees in the Mayan Lab. Today, I'll be talking about a method and software application I worked on this summer to better understand how the transcription factors that are enriched at different time points within time series RNA-seq data regulate each other over time. As a bit of background, ChEA-3 is a tool that enables users to input a gene set and then determine which transcription factors are most enriched for that input set. And to do so, ChEA-3 computes the significance of overlap between the input set and a transcription factor's target gene set in a given library. Specifically for ChEA-3, 6 different libraries were used to generate target gene sets for each transcription factor, including ChIP-seq and gene co-expression libraries. And finally, when determining which transcription factors are the most enriched, the average rank of each transcription factor across all 6 libraries is calculated. Building on ChEA-3, another tool known as ChEA-KG, which was led by Anna Bird, can be used to construct a regulatory network connecting the enriched transcription factors from an input gene set. In this network shown here, red arrows indicate that a transcription factor results in decreased expression of its target transcription factor, while green arrows indicate that a transcription factor results in increased expression of its target. And so with my project, given time series RNA-seq data, we calculated differentially expressed genes, or DEGs, at each time point, and used ChEA-KG to determine how the enriched transcription factors at one time point regulate the enriched transcription factors at the neighboring time point. Specifically, my project looked at time series RNA-seq data, which is relevant because biological processes like tissue development or tumor progression involve gene expression changes over time that can be measured by taking RNA-seq samples at different time points. Therefore, the goal of my project was to visualize the transcription factors that are enriched at different time points in time series RNA-seq data. And by understanding how the enriched transcription factors at one time point regulate future enriched transcription factors, we can better understand how the transcription factor landscape governing a biological process evolves over time. My approach to visualization of time series enriched transcription factors was threefold. First, for every dataset, I constructed a time series regulatory subnetwork to visualize how the enriched transcription factors at every time point regulate the transcription factors at the next time point. Specifically, in the network shown here, the blue nodes correspond to the top 5 enriched transcription factors for the upregulated genes, while the red nodes correspond to the top 5 enriched transcription factors for downregulated genes. Next, I represented the results of the transcription factor enrichment analysis at each time point with a bar chart depicting the average rank of each transcription factor across the ChEA-3 libraries. And finally, I used an animated UMAP plot to cluster each transcription factor based on the target transcription factors it regulates, meaning that points found closer together represent transcription factors that likely regulate similar genes, and this was just a way of identifying potential modules that might work together to dictate some cellular response. I also implemented these analyses into a software application developed using the Apiter platform, which was made by the Mayan Lab to enable users to rapidly develop bioinformatics pipelines. The Apiter that I developed works by enabling users to input a raw RNA-seq gene counts matrix and a sample metadata file describing which time point each sample was taken at. However, users can also directly input a GMT file containing DEGs, and the Apiter will move directly to visualization. But given a raw gene counts matrix, the Apiter uses DSeq2 or characteristic direction to compute the DEGs through 2 different comparison methods. DEGs are found by comparing gene expression at adjacent time points or by comparing gene expression at a given time point with the initial time point. And then finally, the 3 visualizations are run. So now I'll begin discussing example use cases of my Apiter to demonstrate how this analysis can inform hypotheses about biological processes. In the first case study I looked at, researchers found experimentally that induction of triple-negative breast cancer cells with a cytokine called TRAIL led to activation of the non-canonical NF-kappa B2 pathway in the cancer cells, which ultimately resulted in an immunosuppressive tumor microenvironment. In the regulatory subnetwork that's shown here, that was constructed from the researchers' time series RNA-seq data, we see that at early time points, there's an abundance of immediate early genes, which are well characterized to have an early response to extracellular signals like ChEA. These transcription factors include FOS, JUN, and FOSB, which can dimerize to activate the expression of other genes. These early response transcription factors then regulate other transcription factors like ATF3, BHLHE40, and ELF3 that have many incoming edges into them and therefore may play a biologically meaningful role in the response of the breast cancer cells to ChEA. So therefore, from this network, we see that there might be other critical pathways implicated in the response of breast cancer cells to ChEA that are relevant to follow up on. Later on in this network, we see that NF-kappa B2 and its dimerization partner REL-B are amongst the top 5 enriched transcription factors for the upregulated genes at the hour 0 versus hour 12 time point, which aligns with the experimental findings of the researchers. Looking at the network, we also see that PRDM4 and ZNF524 regulate both NF-kappa B2 and REL-B. So therefore, a plausible hypothesis could be that PRDM4 acts as a direct positive regulator of NF-kappa B2 and REL-B, while ZNF524 acts as a direct negative regulator. PRDM4 and ZNF524 are not documented in the literature to regulate NF-kappa B2 or REL-B, so these could act as a hypothesis for novel regulatory mechanisms. Also, from a UMAP plot of the top 10 enriched transcription factors for both up and down genes at each time point, there's a clear cluster of transcription factors corresponding to NF-kappa B2 and REL-B at the hour 0 versus hour 12 time point. However, as circled in the plot, there are other distinct transcription factor modules or clusters that regulate different genes that may be critical in the response of triple-negative breast cancer cells to ChEA. And then lastly, I investigated the most recent GTEx dataset. Adult GTEx is a common fund-supported project that has collected gene expression data from 54 tissue types donated from 1,000 plus deceased individuals spanning different ages and types of death. From this data, I generated sets of aging signatures for each tissue and sex by taking the bulk RNA-seq data from each tissue and using dSeq2 and CD to find the DGs between age groups. Here I focused on the blood from human females, and I noticed that a transcription factor called SPI1, which is a key early regulator of the myeloid and B cell lineages of hematopoiesis, was heavily enriched for the downregulated genes when comparing the 70 to 79 age group to the 20 to 29 age group. In fact, SPI1 was more enriched for the down genes than the up genes at this time point comparison, as seen in the plots below, or here. A literature search also revealed that SPI1 indeed decreases in expression in mouse blood during aging, and in fact, from the GTEx analysis, SPI1 also appears in the down gene sets in the older human age groups. And so altogether, this information supports a hypothesis that a decrease in SPI1 as one ages drives the decreased gene expression observed in aging blood samples. So in conclusion, I show that a combination of all 3 visualizations yields useful information about the regulatory pathways implicated in time-evolving biological processes, such as the response of cancer cells to cytokines or the response of blood cells to aging. This analysis is also available as an appeter that can be found directly on the ChEA KG website at the link shown here. And finally, in the future, we can apply this approach to single-cell RNA-seq data by performing transcription factor enrichment analysis on DEGs found along the pseudotime axis, and we can also develop a scoring algorithm that takes into account the rank of the enriched transcription factors and the number of edges going into each node to prioritize which transcription factors to investigate. Finally, I'd like to acknowledge all the members of the Mayan Lab, in particular Dr. Mayan, for the opportunity to conduct this work and his constant feedback and support throughout my project, and Ms. Sherry Jenkins for coordinating this summer program.